Hello guys. So we are talking about glycolysis. Okay, and we have completed some the reactions of glycolysis at least. So let's go one more time here and let us understand this one more time. What is happening if we talk about glycolysis? So of course we started with, yes, we started with glucose. We started with glucose and we got converted, and we converted this into, okay, let me draw this as a reverse. And this was converted into, yes, glucose 6-phosphate, and this glucose 6-phosphate was converted into fructose 6-phosphate. Okay, this fructose 6-phosphate, okay, here the enzyme was hexokinase and glucokinase. Here the enzyme was, yes, what was the enzyme here? Here the enzyme was phosphoglucokinase. This fructose 6-bisphosphate was converted into fructose 1,6-bisphosphate, and the enzyme here, which is phosphofructokinase type 1, PFK type 1. Okay, in this reaction we have gained one ATP, invested ATP, sorry. And in this reaction we have invested one ATP into ADP, and of course this is also an irreversible step. Okay, this is also irreversible step. After this fructose 1,6-bisphosphate, what happened? You split this thing into DHAP and glyceraldehyde 3-phosphate, and the enzyme was aldolase, correct? Aldolase type 1 and type 2. Of course, DHAP, which will be also converted into glyceraldehyde 3-phosphate. Enzyme here is triose phosphate isomerase. Okay, now this both glyceraldehyde 3-phosphate and this glyceraldehyde 3-phosphate will go into the next section. And now what we are getting here, and this glyceraldehyde 3-phosphate will be converted into 1,3-bisphosphoglycerate, 1,3-bisphosphoglycerate, and the enzyme here which was working very well was glyceraldehyde 3-phosphate dehydrogenase. Okay, glyceraldehyde 3-phosphate dehydrogenase. Okay, and here what we have done, here we have added one inorganic phosphate. Here we have added one inorganic phosphate plus NAD, and what we have got NADH, and this molecule has got one more phosphate group. So we have two molecules of 1,3-bisphosphoglycerate. So of course here also there will be NAD to NADH. Okay, after this 1,3-bisphosphoglycerate, what happens? 1,3-bisphosphoglycerate will be converted into 3-phosphoglycerate. Okay, this will be converted into 3-phosphoglycerate, 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 and the enzyme over here, which is the enzyme over here is forgot the name. This is a kinase enzyme. Okay, and here what we are doing, here we are taking ADP and converting into ATP. So here there is ADP and ATP. Okay, so there is one kinase enzyme working over here, which is phosphoglycerate kinase. I am very sorry, phosphoglycerate kinase. Sometimes my all the bodies using this rapid Lou-Behring cycle, so energy is less sometimes, so I cannot remember properly. But yeah, we have got two ATPs over here. Then when we talk about 3-phosphoglycerate, there is one more mutase enzyme which work, and we get 2-phosphoglycerate. Here also we get 2-phosphoglycerate, and the enzyme over here is phosphoglycerate mutase, Okay, now this 2-phosphoglycerate will be converted into phosphoenol pyruvate, phosphoenol pyruvate, and in from this phosphoenol pyruvate, which is the enzyme over here, the enzyme is enolase. And what we are doing, we are removing water from this. So we have two molecules of phosphoenol pyruvate, and from this phosphoenol pyruvate we will get two molecules of two molecules of pyruvate. And here there is one more ATP to ADP. Yes, we have reached. We have got one more ATP and we have one more ADP. Okay, so this is the overall an idea of our whole glycolysis which we have discussed till now. Now, so what we have done in short, we have taken one glucose and converted into 2 pyruvate. Correct? And in this 2 pyruvate, what we have got? We have invested 2 ATP. Okay, we have invested 2 ATP. We have invested, yes, we have invested 2 ATP. We have also invested 2 NAD. And what we have got? We have got 4 ATP and 2 NADH and also 2 ADP. Correct? 2 ADP we invest किया था. And we have got 4 ATP and 2 NADPH. Not NADPH, sorry, NADH. Okay. So what is the net ATP gain which when we talk about? Here we have invested 2. I want to fit this in your mind very perfectly. Invested 2 ATP, gain of 4 ATP, or you can also call return 4 ATP. So net profit, net ATP gain is 2 ATP. Okay, correct? And we have also got 2 NADH. So NADH is equal to 5 ATP. So in a short, we can also talk about we have net gain of 7 ATP from glycolysis. Okay, we have got 7 ATP of glycolysis. But normally when this will happen, when 7 ATP we will get only when there is mitochondria and there is oxygen. Okay, or at least the enzymes related with oxidative phosphorylation or enzyme related with electron transport chain. Only if these enzymes are there, only then your NADH will be converted into, yes, NADH will be converted into, what is this? ATP, 7 ATP. Okay, so this is overall idea of this. Something about I left about this last reaction, which is phosphoenol pyruvate to pyruvate, and the enzyme here is pyruvate kinase. Okay, what happens is, you know, sometimes pyruvate kinase is mutated, or let's say any of the enzyme, any of the main enzyme is mutated, let's say specifically in RBC. Okay, now you say RBC mein how it is mutated? See, RBC gets its all gene when it is in erythroblast. Erythroblast will synthesize and transcribe and translate all the genes, correct? So this erythroblast, when it will convert into RBC, it will always have its all the genes, all the proteins which are made in the previous stage, which is in erythroblast stage. Okay. So of course, when we talk about erythrocyte or RBC do not have your citric acid cycle because they don't have mitochondria and they do not have they have oxygen but they do not have mitochondria, so the citric acid cycle and electron transport chain will not be run properly. So you will have, see here what we are telling, when we are talking about normal cells, normal eukaryotic cells, these cells will have citric acid cycle and this NADH will be used, correct? And this NADH will be converted into NAD in those citric acid cycle, and in your specifically in electron transport chain, this NADH will be converted into NAD. But in RBC there is no citric acid cycle, so what will happen to it? So in RBC specifically what happens is, RBC, your pyruvate is converted into, see normally what happens, the pyruvate can go into, okay, the pyruvate can go into your citric acid cycle. Of course the pyruvate has to convert into acetyl CoA. Acetyl CoA is 2 carbon molecule, pyruvate is 3 carbon molecule. So one carbon dioxide molecule, carbon molecule will be puffed out as a carbon dioxide. Okay, so this pyruvate in RBC, this will not happen. This pyruvate will be converted into lactate, and the enzyme here is lactate dehydrogenase, and the enzyme over here is lactate dehydrogenase, and in this lactate dehydrogenase you can use NAD is used and we get NADH, and this NADH will be used over here. So you can understand now, if you don't have, see in this I'm very sorry, ulta bol diya maine, NADH will be used and you will get NAD. Okay, here NADH will be used and it will be converted to NAD. Your lactate dehydrogenase enzyme will take NADH and it will convert into NAD, and this NAD will be used for this reaction. You can understand, if there is no citric acid cycle, NADH will be not converted into NAD. So here NAD kahan pe milega? So if NAD is not here, the glycolysis will stop. That's why we have NADH will be converted to NAD and NAD will be regenerated here, and this NAD will be used in this glyceraldehyde 3-phosphate to 1,3-bis phosphoglycerol. Okay, so sometimes what happens, and see the main demand of RBC is not very much ATP. So RBC normally has this kind of pumps, correct? RBC has this kind of sodium potassium ATPases which transfer sodium and potassium inside and sodium outside, 3 sodium out and potassium, and it works on ATP. So majority of the ATP of originated from glycolysis is used in your sodium potassium ATPases. Okay, so if any of the enzymes, let's say pyruvate kinase, most of the cases the pyruvate kinase is mutant, or it is not made properly, or the affinity towards the phosphoenol pyruvate is very less. So if this pyruvate kinase enzyme, okay, this pyruvate kinase enzyme, if it is mutated in your erythroblast and it's appear in mutated pyruvate kinases appeared in RBC, so what will happen? So if it is mutated, so the energy will not be made. Why? Because this step will not go, phosphoenol pyruvate will be not converted into pyruvate. That's why the glycolysis will somehow stop. If ATP is less made, sodium potassium ATPases will not be made. Sodium potassium ATPases will not work. So if sodium potassium ATPases will not work, sodium will remain in the RBC. There will be lot of accumulation of sodium in the RBC. If sodium is accumulated in the RBC, who follows sodium? Water. If water is coming into RBC, this RBC will be in this shape, spherical shape. Okay, it will be little bit swelled, bulgy. It will not be very nice shape molecule. So we have to understand what we are talking about, because this shape is very important. Okay, because this shape is very important. Why? Because specifically this shape is tested in the इसका main exam होता है spleen में, because in spleen you have very small capillaries, and in that capillaries this RBC bends and it passes away. It bends and it makes some turns and it passes away because of the shape, because of the biconcave shape. Okay, if it can bend only it can pass. Okay, so whoever bends can pass through that spleen capillary and it can survive. But do you think this RBC will able to do that? So this RBC will not be able to do that. So this RBC will be trapped into the spleen. If this RBC is trapped into the spleen, all the macrophages will come and start eating, the RBC, and that's why the RBC will prematurely die. Okay, so if RBC is very less, so of course there will be lots of hemolysis, and because of this premature hemolysis, see, normally RBC lives for some 2 months, 120 days or something like that. Okay, and this after their life cycle, after the lifespan is completed, they die, and then macrophages eats them and everything happens. But this RBC goes into the spleen and die prematurely. Okay, and that's why the rate of RBC goes down. If rate of RBC, because of this premature hemolysis happens, okay, this premature hemolysis happens and the person might be anemic. Okay, the person might be anemic. Are you understanding what I am saying? So this hemolysis happens for many reasons, but premature hemolysis happens also for many reasons. So one of the reasons is this thing. Specifically in glycolysis, there are two molecules which or two enzymes which, when it is deficient, it can cause problem. One is glucose 6. One is our pyruvate kinase for sure, and other is our dehydrogenase glyceraldehyde phosphate dehydrogenase enzyme. When they are mutated, this problem happens because everything stops. Okay, so this is some points about So other thing, when we talk about this glycolysis, let us talk about a little bit about glycolysis. So this glycolysis is of two types. One is aerobic glycolysis and one is anaerobic glycolysis. Aerobic, and let me tell you what do I mean by that. By the name itself you can understand what is aerobic and what is anaerobic. Aerobic means it needs oxygen, and anaerobic means it doesn't need oxygen. Okay. If you talk about aerobic glycolysis, okay, if you talk about aerobic glycolysis, you have a mitochondria. This glycolysis means, what is the glycolysis? Glucose is converted into pyruvate. Okay, this pyruvate can go into mitochondria. Okay, this can go into the mitochondria and form acetyl CoA. Okay, and this acetyl CoA can go into citric acid cycle, and after this citric acid cycle, some of the NADH and FADH, it can go into electron transport chain, and this electron transport chain can give you lots of energy. So one fate of pyruvate goes into your mitochondria, and here we talk about the ATP gain. If we talk about this cycle, ATP gain here is 7 ATPs, correct? Because 2 NADH will be converted into 5 ATPs in mitochondria. So this is one fate of pyruvate which is going into acetyl CoA stuff. Okay, so this is an aerobic glycolysis. When we talk about pyruvate has some more fates. For example, once we have talked about in RBC, that in RBC or in the cells which do not have mitochondrial oxygen, this pyruvate can be converted into lactate. This pyruvate can be converted to lactate, and the enzyme here is lactate dehydrogenase. Okay. This pyruvate can be converted into lactate, and this lactate can happen, and this lactate can be very useful for other stuff, correct? And the enzyme here is, because it is also taking NADH, and it is also taking NADH and giving to NAD. Okay, so one of the fate is your lactate or this pyruvate. Some of the yeast cells. Okay, if you talk about some of the yeast cells, these yeast cells can take this pyruvate. Thank God we don't have this pathway in our body. Some of the yeast, some of the plant cells also, some of the plant cells, some of the yeast cell can take this pyruvate and convert into ethanol. Convert into ethanol. And this ethanol will be also using NADH and giving NAD. In this NAD will be used over here in this reaction. Okay, so here we talk about fermentation. So fermentation, what do you mean of fermentation? Anything happens to the glycolysis. Okay, the glycolysis is the pathway which happens, specifically the anaerobic glycolysis which we are talking about, that is fermentation. Okay, that means glycolysis under anaerobic condition. Okay, the end product can be lactate or ethanol, that is fermentation for you guys. Yes. Some more stuff we will see. We'll go a little bit in more detail about this fate of pyruvate. See, one thing I have forgot, I just remember that we have talked about that the pyruvate and alanine also, in last lecture I have talked about alanine also inhibits pyruvate kinase, because this pyruvate can be converted into alanine very easily. If you see pyruvate structure and alanine structure, it is almost similar. Just you have to add one NH3 group. Okay. So this, if alanine is more, you don't need more pyruvate, so it will stop the pyruvate kinase to make more pyruvate. Okay. Specifically, when we talk about, let's talk about some of physiology of glycolysis. In our body, most of the tissue runs aerobic glycolysis, which is from mitochondria and everything. Okay, but there are some cells which runs anaerobic glycolysis, that is our RBC, lenses. Okay, the lens in the eye, some part of kidney, specifically in renal medulla. Okay, they run anaerobic glycolysis. And any tissue which is in heavy use of glucose, for example our muscle cells, when our muscle cell we are exercising heavily, what happens, the glucose demand is not met by our exercising needs. That means we are exercising a lot, that means we need lots and lots. So glycolysis will run very fast. Okay, if glycolysis is run very fast, oxygen is not coming that in high rate. If oxygen is not coming in that high rate, because there is a limit of the speed of what we can deliver oxygen to places. So of course there will be hypoxia in that region. If there is hypoxia in the region, so this pyruvate will not go into acetyl-CoA pathway, it will go into lactate pathway, and this lactate will start accumulating in our muscles and whichever tissue is using or running the glycolysis. So this lactate or lactic acid will be taken into, okay, so this lactate or lactic acid will be taken into blood circulation, and this lactate or lactic acid will be taken into the liver, and this liver can take this lactate and again convert it back into pyruvate very easily. Our liver can do that, correct? So when we talk about severe hypoxia, in severe hypoxia also this happens. In severe hypoxia, most of the pyruvate will be converted into lactate, and if it is converted into lactate or lactic acid, if the lactate is going in very higher amount, if it is accumulating a lot, that will kind of change the pH of blood, and that will be a problem. And this thing is called lactic acidosis. Okay, this lactic acidosis is not a very good news. And this can cause many problematic things like severe hypoxia. Will lead, see, severe hypoxia can lead to many, or can be lead because of many reasons. For example, severe hypoxia or generalized deficiency of oxygen in every tissue can achieve through many reasons. For example, there is myocardial infarction. If there is myocardial infarction or some kind of heart attack, in that kind of what happens, our body is going under circulatory shock. If it is going under circulatory shock, all the different body parts will not get oxygen in that amount. So if it is not getting oxygen in that amount, so of course our pyruvate will be converted into lactate, and there will be lactic acidosis. Okay. Then the other reason will be, let's say if there is any thrombus or some clot in the heart, again the circulation is stopped. Okay, if there is severe accident or hemorrhage in our body and there is heavy blood loss, if there is heavy blood loss, of course RBC will be less and there will be generalized hypoxia. In this generalized hypoxia, what is happening? Your blood pH is altered. There is the case of lactic acidosis. Okay, there is a whole lot of problematic things which happens. Okay. And of course this pyruvate will be converted into, it doesn't happen to us, but in microbes, pyruvate is converted into acetaldehyde, and acetaldehyde is converted into ethanol. Okay. Yes. I guess we should see this reaction little bit in detail because थोड़ा ये रिएक्शन थोड़ा सा डिटेल में देखना चाहिए. This is very important. We talk about fermentation. People talk a lot about fermentation, but you don't understand what is fermentation. Correct? So when we talk about anaerobic glycolysis, there are two things. One is conversion of your pyruvate to lactate. So you take pyruvate. And of course you take pyruvate and you also take NADH we have got from this reaction. You take pyruvate and NADH and then you convert into lactate. And of course this reaction is reversible. Okay, enzyme here is lactate dehydrogenase. Okay, this lactate plus NAD of course, and this NAD is utilized over in this glycolysis step. Okay, so here what is happening? Here pyruvate is reduced back to lactate. Okay, so that NADH can be oxidized. NADH can be oxidized into NADH and pyruvate is reduced into the, see, you will understand when I will show you the structure. Okay, let's say this is our pyruvate. So simply ऐसा ही लिखते हैं. CO minus and you have one double bond. So this is your pyruvate and you can see the lactate structure. Okay. So what we have done? We have done the reduction. Pyruvate का reduction हो गया है, या फिर we can also call it hydrogenation. That means we have attached hydrogen at some point, and what has happened to our NADH is going into oxidation. That means NADH का बन गया NAD. That means उसका हो गया dehydrogenation. Normally hydrogenation happens in reduction and dehydrogenation happens in oxidation. Okay, so this is your thing. And the second step we were talking about in anaerobic glycolysis, that was conversion of pyruvate into ethanol. Okay, which is alcoholic fermentation. So you have pyruvate. That pyruvate will be converted into, I will not give the structure. it's not important. This pyruvate will be converted to acetaldehyde, and this acetaldehyde will be converted into ethanol. Okay, it will convert into ethanol, and this ethanol, of course, this step, if you talk about the acetaldehyde to ethanol, it takes NADH and it converts into NAD, and this is called alcoholic fermentation. And of course, yeah, ethanol, if you talk about ethanol, ethanol is 2 carbon, if I am not wrong. Ethanol is 2 carbon long. Okay, ethanol is 2 carbon. Ethanol ka structure bhul gaya. Sometimes it happens to me that we don't remember ethanol structure. So this is ethanol, 2 carbon compound. So one carbon dioxide. So one carbon is released in form of carbon dioxide here. One carbon is released in form of carbon dioxide here. And it is going out. Okay. That's why if you see many fermented foods, you will see this step. The ferment, like idli, dosa, it becomes spongy and everything. And this sponginess and this fluffiness is because of this carbon dioxide which is getting released from the alcohol fermentation. Okay, and of course the third thing is our aerobic, where pyruvate will go in acetyl pair. That we will see into the next when we study citric acid cycle. Okay, and linking step. Thank you very much.